So guys, again, I'm glad we got a chance to gather, even though we're in the midst of this global coronavirus pandemic. We got to keep our spirits up while we're inside and keep our learning going. So today I'm going to talk to you guys about viruses. I'm going to give you guys a lecture. It's actually a lecture that I've delivered to undergraduate and graduate students at Howard University. So this is university level material. I feel that you guys having now gone through our Future Kings biomed training program, you guys have the background skills and capability and understanding of molecular and cellular biology to begin to follow this. So I'm going to go ahead and share my screen, and then you guys can follow along. And throughout the lecture, if you have questions, just unmute yourself and ask them. Okay, feel free to interact, and we'll go through maybe about 30 minutes of the lecture. I'll try to get through it in 30 minutes, and then we'll have plenty of time for questions. And then at the end, we're going to learn about coronavirus, the actual virus that we're experiencing right now as a pandemic, and set up our future sessions where we're going to brainstorm on ways that we could use our skills in molecular biology to try to come up with new treatments or therapies for coronavirus. Okay, so let me share my screen and get going. Okay, let me make this bigger. I gotta go all the way to the beginning of our lecture. Okay. Okay, so hang on, let me kind of minimize that. Can you guys see? Okay, good, we're good. All right. So what is a virus? So when we think of what a virus is, it is essentially what we call an obligate intracellular parasite. That's a big, long way to say it's something that likes to get inside and live off of our cells. So fundamentally, in a genetic sense, a virus is a package of genetic information that's protected by a protein shell for delivery into a host cell whose sole purpose is to be expressed and replicated. So there's been a lot of arguments in history, is a virus a life form? Well, because it can't really exist on its own, I don't think it is. That's my personal opinion. Because a virus, one of the key hallmark principles of a virus is that it needs a host cell in order to make more of itself. It is the ultimate parasite. And so that's why we call it an obligate intracellular parasite, something that needs to be inside the cell in order to live. And so, question?
No, I just was hoping I didn't drop you out. Okay.
No, we're good.
Okay.
So there, viruses. There are a lot of other kinds of parasites in the world, and you've got different kinds of organisms that are even simpler than viruses, like mycoplasma. These are small bacteria that grow on medium and attach to cells. These actually, when you work in the lab, mycoplasma is a common contaminant of cell culture. It's a real pain in the neck for people in the lab because they're these tiny little things that just love to grow in media. Chlamydia is a bacterial parasite which depends on cells for energy, and it causes some sexually transmitted diseases. Protozoans, these are parasites that replicate inside of eukaryotic cells. And then there are these new things called viroids. These are like infectious agents of plants that are kind of just naked nucleic acid. That is, they don't have the protein coat. They just have a single-stranded RNA that makes them infect plants. And then you have these virus-like things. So this thing's called hepatitis delta virus. They call it a viroid-like agent. That it's dependent. Its replication is dependent on hepatitis B virus, right? So you've got a virus who's dependent on a virus. So like that's the ultimate parasite. But for all intents and purposes, all of the viruses that infect us, have been around for a long time. So how long have we known about them? So you can trace viruses all the way back to maybe around 1500 BC, where they saw leg deformities that looked like what we would normally call in the modern day polio or poliomyelitis. And then you also had pox marks that were hallmarks of smallpox viruses. So up to 1500 BC, back in ancient Egypt and other places, you saw these things, and these were being described by some of the very first physicians. So viruses have been around for a very long time.
Hey, Dr. Cunningham, I have a
Yeah,
question.
go ahead, Gideon. Yeah.
So will we associate viruses to be bad? I remember we talked about this, but I forgot what was the answer.
So viruses themselves aren't necessarily bad. I would call them just a part of nature. They're parasites. So for the organism that they infect, they can be bad, just like we're experiencing with the people that die from viruses. But as I'm going to talk later in the lecture, the goal of the virus is not to kill the host, right? Because if you're a parasite, you really want your host to live, because if the host dies, you die, right? So the goal of a virus is not to kill the host. So that's a great question, Gideon. But I think, and again, since viruses can't live on their own, they're just they require our cells in order to live. They really need to try to keep us alive. Now, the viruses that kill us are kind of doing it by accident, right? Because that's not really to their benefit to kill us. So COVID, HIV, smallpox, these viruses that kill us is because they really haven't worked out the correct symbiotic relationship with us as the host yet. But that's a great question.
Is there a virus that we benefit though? Like we can benefit from?
That's a good question. I think honestly, ancient viruses actually are in our DNA. Much of our DNA is actually made up of ancient viruses that were called retroviruses, right? Very similar to the modern day HIV. And these are actually the way that we change over time. So these viruses have now become a part of us. They're called endogenous retroviruses, and they live within us. And there may be some positive things that they do. We still don't understand all of the good stuff. But let me get back to what I'm talking about. So even in Egyptian mummies, they found evidence of smallpox, right? So mummies from, almost 2,000 years ago had these things as sort of the first indications of smallpox, of viruses. And virus, the word, comes from the Greek term that means poison, right? And it was initially described by Edward Jenner in 1798. Do any of you guys know who Edward Jenner was without me even saying it?
I've heard of him.
Who was he?
Was he the guy who did the vaccine?
That's right. He's the first guy to do vaccination. He actually coined that term. And he was famous for doing the first protective vaccine trial where he took cowpox. He took exactly. My son Logan is in the background. He's telling the story. Edward Jenner was the first to take inoculate people with cowpox and protect them from smallpox. So it's kind of amazing. And that happened in 1798. So this is, 222 years ago, right? That we've only been able to do that. So it's kind of amazing that we have now thousands of vaccines for many different kinds of viral infections, or hundreds rather, not quite thousands yet. Okay. And then, but let's kind of understand where we've come. So from the 1800s, all infectious agents were thought to be viruses, even if they were bacteria, because back then they didn't really know. They didn't really have great microscopes back then, so they couldn't really see, the difference between a virus and a bacteria. So there was a guy named Robert Koch who developed these culture techniques, which allowed you to separate bacteria on growth media. And in these 1800s, the later 1800s, you were able to purify these agents with filters. So you kept using a more and more refined filter that made smaller and smaller filtration in order to filter out bacteria versus viruses, because bacteria are bigger than viruses. They're a lot bigger. I'll show you guys the scale. So some of the first viruses that were described were called foot and mouth disease. And then in 1898, yellow fever. And then in 1900, there was a virus that they discovered called Rouse sarcoma virus that actually causes cancer. And there are certain viruses in animals and even in humans that can actually cause cancer because they integrate into the DNA of the host and they can cause mutations. So that's kind of amazing. And Peyton Rouse, who discovered that virus, won the Nobel Prize for it 114 years ago in 1906. Okay. So these viruses have played a major role in human history. And whatever time scale you want to look at, over the past 1,000 years, smallpox and measles came to North and South America, and they were killing European explorers and conquerors, as well as the indigenous populations. The native populations were really crushed by these viruses because they had no immunity. And so our major defense against viruses as humans is our immune system. So it's always a race between the virus that's infecting a given organism, including humans, and that organism's immune system as to whether or not that will determine as to whether or not that organism survives. So over the past 100 years, we've made a lot of progress. But to put everything in perspective, about 102 years ago now, there was a newly emerged strain of influenza, right, that killed 50 million people in 1918, between 1918 and 1919, immediately after World War I. Oh, they called it the Great Influenza. It was the influenza pandemic. And then not even 10 years later, polio became one of the worst diseases in kids. It was causing crippling disease in children. So these were really important viral pathogens that just even 100 years ago were causing major problems in this country. Even the president, Franklin Roosevelt, was a polio sufferer. That's why he lost the use of his legs and had to be in a wheelchair because he fought that disease. So these viruses have affected us over many years. And then now in this century, in our last, let's say 30 years, right? 40 years from the 1980s to now, you've had HIV, which have infected over 75 million people and killed 32 million. And then you've had multiple outbreaks of SARS and influenza, right? So you had SARS, you had MERS, and now the current COVID-19 that are causing a lot of problems throughout the world. Any questions, real quick?
I have a question.
Mm-hmm.
So I know that around 30 years ago, around like in the 80s, people thought that homosexuals transmit HIV. I don't understand why they thought that.
Well, they were the first populations where it was spreading in. So that is true. Like, so back in the late 70s, actually, the HIV epidemic was first identified in mostly homosexual or gay men that had the disease, and they were spreading it like really fast because they were having, like unprotected sex and doing unsafe practices. So what happened was they started seeing these clusters, and it was really devastating this particular community, but it wound up being spread everywhere. I mean, AIDS actually had the highest death toll in Africa among people who were heterosexual, folks who were just passing it on through sexual contact, unsafe practices. So while in the US and in Europe, it emerged in those populations first, it's actually a disease that can infect anybody. And HIV is a good example of a disease that modern medicine has really attacked. And my PhD thesis advisor was one of the first physicians to see HIV in Los Angeles when he was much younger. And he was one of the first people to witness it. And as a result, he was one of the first scientists to study it. And he and his research team came up with the drug cocktail that has now saved millions of lives. So they came up with the whole concept of treating the virus with multiple different drugs to try to block it at multiple different stages. That was a really important principle. And the information and the science that we gained from fighting HIV were now being applied to fighting coronavirus. And I just literally read an article last night that my former thesis advisor, Dr. Ho, is now working on coronavirus using these same methods. So I'm hoping that his research team can find some breakthroughs fast. So in the last, I would say, 20 years, right, you've had HIV and now Ebola, which is another kind of virus. This is actually a picture of Ebola. This is what Ebola looks like. Many viruses look like round, right? But Ebola looks like a filamented virus. It's called a filovirus because it looks like a filament, like a long string, right? But this is an electron micrograph. So it's like we're viewing the virus at 160,000 times normal magnification. So this is what viruses look like. That's how small they are. We have to look at them on an electron microscope, right? And in order to get a look at what this does, all this thing does is make more copies of itself. it gets into our cells and it makes more copies of ourselves. And in the case of Ebola virus, the reason why it kills us is kind of crazy. It makes one protein on the outside of its body, of the body of the virus, called the envelope protein, the S-glycoprotein of that virus. And what that protein does is it inadvertently, by accident, pokes holes in the cells that make our blood vessels. Okay, so what happens when you poke holes in the cells that make your blood vessels? What do you think will happen?
Can't take as much
It
off.
explodes.
What will happen? If I poked holes in your blood vessels, what would happen to your blood?
It start to leak out.
It would leak out.
Yeah.
And so that's how you die from Ebola. You start bleeding internally, and you literally bleed. In the worst cases, people will even bleed from their eyes, right? Because the vessels literally are bursted, and then you lose your blood. And if you lose blood, your tissues can't get energy, because that's how your tissues get energy, from the oxygen from your blood, and then you die. And it's a really horrible way to die, painful. And Ebola kills about 30% of people that it affects. Question?
What I don't understand is that how come when people get treated for Ebola, sometimes their eye color turns to a different color.
Right. So that's the blood. That's the actual bilirubin in the blood being broken down from all the blood in your body being outside of your vessels. You'll get breakdown of bilirubin, and your eyes will turn like yellowish or really dark colored, because you're also busting all of the tiny capillaries in your eyeballs with these proteins. So it's a really horrible, horrible way to die.
Is that?
And-
Do you can't you heal from that, or no?
Yes. So 60% of people that get infected will get an immune response that will stop the virus, right? So you have to. The reason when you get infected with a virus, you feel sick, right? It's because your immune system is trying to fight it, right? That feeling of being sick, that feeling of having a fever, that's your immune cells kicking into action and trying to go and fight the virus, right? And so you need some time in order to be able to generate both an antibody response and what's called a cell-mediated response, actually immune cells that go and hunt and kill cells that are infected with the virus, so that you can clear the infection, right? So this modern discipline of virology, it's only really in the last, I would say, 100 years that we've had it. Even though Edward Jenner did the first vaccination way back when, and right before 1800, really understanding what a virus was has really only happened, I would say, even in the last 60 years, that we've been able to look at them really carefully on microscopes and then actually manipulate them at the genetic level. Okay. So, these disciplines of virology is kind of a mixture of infectious diseases and molecular biology, because we can use the principles that you guys have been learning, like PCR and how to amplify DNA, and also RNA. You guys have learned these techniques, and we use these in the lab to actually understand what these viruses are. So when they're trying to understand when a new virus is emerging, they use PCR, right? The technique that you guys learned in the lab, they use PCR, and they use it based on viruses that they know. So for example, this new coronavirus is called SARS coronavirus 2019, or SARS-2. A lot of researchers are calling it SARS-2 because it looks kind of similar to SARS-1 at the genetic level. It's about 80% similar, right? So we use these disciplines of infectious disease and molecular biology to help us understand viruses. So I wanted to highlight this guy, Koch, because he gets a lot of credit for really proving Louis Pasteur's germ theory of disease. So Louis Pasteur was one of the first people to really recognize that germs, or things like bacteria and viruses out there, actually cause disease, right? Because back in the day, people used to just get sick and they didn't know what was going on, right? They were like, oh, you just got sick because you couldn't see anything, you didn't know anything, what was happening. But Louis Pasteur came up with the concept of the germ theory of disease, which led to things like pasteurization, right? So milk used to have tons of bacteria in it, right? And Louis Pasteur came up with a technique to basically boil the milk and kill the bacteria in it. And that's why we all now drink pasteurized milk, so we don't have bacteria.
Was this the same guy who discovered spontaneous generation, or was that a different guy?
You might be thinking about a different guy.
Yeah.
What Koch did here, he developed a series of rules called Koch's postulates, and these are what define what a virus infection is, okay? And I'm going to send you guys this lecture. This is actually a college level lecture, so I have a quiz and everything on it, but I'm just sending you this, that. So these are rules for being an infectious disease from Koch, right? It's number one is that the microorganism, whatever it is, a bacteria or a virus, must regularly be able to be isolated from people who have the disease. So in the case of smallpox, you could take one of the pus blisters from smallpox and isolate the virus out of it, right? In the case of coronavirus, you could take a swab from the nasal or throat passages and isolate the virus out of it, right? And then you must be able to grow it in some sort of culture that is on cells that the virus likes to infect in vitro. That means inside of a dish, in a dish inside of a lab. You can be able to grow it, okay? All right? And then once you've been able to grow it, right, grow it up, then you can take those grown up microorganisms and then put them back into the animal host, be it a human or whatever that's infectable by that virus, and then to see the disease, right? So whatever you've isolated from a diseased infected person and then grown up separately and then reintroduced into a healthy person and then caused the disease, then that's it's fulfilling Koch's postulates. And then you must be able to re-isolate it from then on and pass it on. So it's really simple, right? But these are like these are the rules for what define something being an infectious disease. Something like cancer is not necessarily an infectious disease. It's more of a genetic disease. You have a genetic mutation, and it can cause your cells to grow out of control. But like a virus is an infectious disease because it can pass from person to person, and you can pass it from a sick person to a healthy person and make that healthy person sick. Got it?
Doctor, can I have a question?
Yep.
So for an infectious disease, why did they take samples out of the nasal cavity?
Because that's where the cells in your nasal cavity is where the first place where these viruses get in. When it's an airborne virus, when you inhale air, the first place that the virus latches onto will be the cells that line your nasal epithelium and the back of your throat. These things are called epithelial cells. And then they get in and they start the infection. And then in the case of coronavirus, it spreads from your upper airway to your lower airway into your lungs. And once it gets into your lungs, it can get very dangerous. So we can cultivate viruses on cells, right? So like you guys haven't seen this yet, but this was what I was planning for the second half of our training. But hopefully when we're able to gather again, I'll be able to take you into the lab and see how we culture cells in a tissue culture dish. And we use something called a hood, a biosafety cabinet or a hood. And what that does is it has air pressure in it that takes all of the bacteria and stuff out of the air so that it doesn't get onto your cells. And so it creates a sterile environment for us to be able to culture cells. And this is how we grow viruses. We grow viruses on cells, right? And then in some cases, like flu, you can actually grow virus in chicken eggs, in the yolk of a chicken egg. That's how they actually make current flu vaccines. They take some chicken eggs, they inoculate them with the virus, and then they grow it up, and then they process the yolk and take the virus out of the yolk, treat it with formaldehyde to kill it, and then that's what gets injected into your arm for your flu vaccine. So you're getting injected with dead versions of the flu virus that have been grown up in chicken eggs, right? And then we can also grow viruses in lab animals. You can put them in rabbits, mice, rats. We have these animal models to try to understand how virus infections work. And when we do trials of vaccines, we often do them in animals to see if we can cure the animals and give us some insight on how we could cure humans. Okay, this is just showing how you can inoculate a chicken egg for flu viruses, but different kinds of viruses can be inoculated into chicken eggs, like mumps virus. There's another one called Newcastle disease virus that affects sheep and livestock that's kind of flu-like. You've got all these different kind of viruses out there. Okay, and this is literally what it looks like. You take a virus, you just you take an egg, you cut off the top of it, you inject some virus, and then you put the top back on, and you keep it like in a warm environment, just like a chicken egg. And that's how they actually grow up the flu vaccine.
I have a question.
Yep.
So when these vaccines are made, are there is their purpose to like give the body, I don't know, like an example of the virus? Like,
that's exactly what they're meant to do. So a vaccine, that's what Jenner did so well. He took something that looked a lot like smallpox, injected it into the body, so that the antibodies that people made to that virus could then protect against the virus that it looked a lot like, right? So that's the same thing with the flu vaccine. So they take the flu vaccine. This is how they make it every year. They'll take different strains of the flu and they'll make an estimation based on the sequence now, the genetic sequence of the flu that's circulating. And they'll make a cocktail of flu viruses, generally 3 or 4 flu viruses. They'll inject them into this chicken egg and then get out a mixture of flu viruses, okay? That's the vaccine stock, right? And then they'll purify it from the yolk of that egg. And they do it in millions of eggs. Imagine facilities that have millions of these eggs, and it makes enough to vaccinate the entire population, right? And the idea behind it is to be able to introduce into your body something that looks a lot like the flu virus that you have a high probability of seeing in nature. Does that make sense?
Yeah.
Yep, that's exactly right. The vaccine is meant to trick your body into developing an immune response so that immune response is there before you even see the virus next in real life. Okay, so but you can grow smallpox in eggs too, right? So you get when you take smallpox virus, put it in an egg, it gives you pox almost that looks like on a human, and this is actually smallpox. What a really severe case of smallpox looks like. Now, smallpox had been eradicated.
Yikes!
First viruses to be eradicated from the human population. And they did that through a massive vaccination program. So it took about 200 years later after Jenner did the first vaccination, but within 200 years, we eliminated a virus that had been killing us for hundreds of thousands of years. So that's actually pretty good. It's a great demonstration of how modern medicine can actually impact something. And a virus that kills us is something that we want to actually make extinct. Right? So
Wait, I have a question real quick.
Yeah. What's your question? Yeah.
Can in a laboratory, can we accidentally make a virus, like a man-made virus?
We can absolutely accidentally make it, and we can actually do it on purpose. I'm going to show you how I do that, or I've done that. But I'm making versions of a virus to try to cure it, right? So there's always the chance, and this is the danger with being able to engineer viruses, that you could make a version that was bad. So you'd have like a outbreak that would be unexpectedly lethal. So that's why virologists are very careful and work in very contained environments, particularly when they're dealing with pathogens that can kill people, right? So like when I had to work with smallpox and influenza, I had to get specialized training in biosafety level 3 laboratories, right? Where you have to wear a respirator and go in. The top level is biosafety level 4, which is where Ebola and other viruses are worked with, because they're so deadly, you really cannot get them on you. If you have any, if you get a little bit of Ebola on you, you will get it and you could die. You have a 30% chance of dying, right? So I used to work with smallpox, and in order to work with it, I had to be vaccinated against it. So I was one of the few folks actually that was vaccinated against smallpox as an adult, because I was born after 1970. So they stopped vaccinating kids against smallpox in 1970 because the WHO, the World Health Organization, had basically declared that smallpox had been eradicated in the human population. And it pretty much had. It was only available in labs. So the only people that really needed the vaccination were people like me and my lab mates who were actually doing research with the virus.
What's the difference between smallpox and chickenpox?
They're a different virus. They're the same family, but they're different forms of that virus. Okay, there's a called pox virus or vaccinia viruses, right? Mm-hmm. And so you can look at, we made smallpox vaccines. We can grow it in humans, and then we measure these things, viruses, by their ability to actually form what's called plaques in cells. So when we make a cell, and when we quantify virus, we do it in a way they're called plaque forming units. That's the infectious disease label that we use. So when you hear PFU, it means plaque forming unit, right? And it's the measure of the number of virus particles that are capable of forming a plaque or killing a cell on a tissue culture plate. Like when we put it in a plate on a lab, we can then add the virus to it, and then we can count the areas of where the virus is killing it and estimate how many viruses we have in the solution, right? And so this was some things where I'm going to go through this quickly, but just to show you, right? These are different cell cultures that we can culture viruses in. And imagine these are your cells, right? I tried to kind of make a diagram here of this is what a cell, a layer of cells would look like on a tissue culture plate. When you add your virus, you start seeing, add one virus particle, and then it starts to replicate itself. And then it replicates itself so much that it bursts open the cells, called lysis or lytic spread, right? And so this is what coronavirus is doing to the lung epithelial cells. It's killing them, and that's why people are getting these horrible lung pneumonias, because without your lung epithelial cells, your lungs basically turn to paper mache, right? So you keep getting this killing, and it spreads, and it becomes a plaque. And this is what it actually looks like in a culture. So this is a typical culture of different amounts of virus. So this is like 100% virus, 10% virus, 1%. So you can calculate how many particles of virus you have based on the plaque forming units, literally counting all these dots.
Wait, do you count it manually, or is there like a database you use?
You can count it manually with your eye. You get trained to do that. I'm trained to do that. But you can also now count them with computers. That makes it a lot easier. So you take a picture and it actually counts the plaques. Okay? So this is just some more stuff. You guys will get this when you get like more advanced virology, but I'll skip through it for now. But I wanted to kind of give you guys an example of the size of things, right? So just to give you an example, I told you a bacteria is much bigger than a virus, okay? So remember that. Bacteria infections, even though they're really small and we can't see them with the naked eye, the average bacteria is about 1 micron in size unit, right? 1 micrometer, right? It's micrometer. It's like a thousandth of a meter or a millionth of a meter, something like that. And then viruses are nanometers. So yeah, micrometers are millionth of a meter. Viruses are in the nanometer range. That's billionth of a meter, right? Super, super small. You cannot see them with the naked eye. So the smallest viruses tend to be anywhere from 20 to 200 nanometers in diameter. So pox viruses are the biggest viruses out there. And they have a lot of genes. They have about 200 different genes. And they are almost as big as a chlamydia type of thing that can infect bacteria. But definitely, you know, no virus is ever going to be as big as a bacteria. Okay? And then, so if you look at our ability to see these things, so the relative sizes of things, right? So our cells are, you know, about 10 microns, right? Bacteria, about 1 micron, right? One micrometer. Viruses are in the nanometer range, right? Either between 10 to the 7th, like pox virus, or 10 to the 8th to 10 to the 9th. And then you start getting down to the level of looking at individual proteins, which are nanometer size, and then atoms, which are sub-nanometer, femtometer at the atomic scale, or you'll see atomic resolution, right? So these are really, really small units. And these are the different techniques you can use to see them. On a microscope, you can barely see a large virus, like a pox virus. But on an electron microscope, you can pretty much see all kinds of viruses and proteins. To be able to see atoms, you need to do X-rays and nuclear magnetic resonance, NMR, to be able to see this. So these are really more advanced techniques that require actual radiation in order to see it, right? So once you get down to super small size, you need very specialized techniques on how to view these things. Okay, so I'm gonna kind of go through the different kinds of viruses. So these are some common viral infections. So any questions though, real quick, before I start talking to you guys about the families of viruses and stuff? No? Okay, so there are a lot of common viral infections that we experience like every year, and all of us have had these. There are basically 3 types of viruses that can cause the common cold, right? They're called rhinoviruses, adenoviruses, or coronaviruses. And then also another kind of virus that can cause cold-like symptoms is called the influenza virus or flu, right? Then you get these sort of stomach bugs, right? Which are not necessarily flu viruses or cold viruses. They're actually viruses called rotaviruses or noroviruses, like Norwalk virus and others. Then you have different kinds of viruses that cause cold sores, like herpes viruses.
This
And
is going
real quick,
crazy
question.
until I get. Ooh, thank
Yeah.
you.
So with
Right
herpes
now we're trying to have
viruses,
2 meetings at the same time,
okay.
and it's not letting me. And the guys are sitting there waiting for it.
All right. Hey, Eric, we can hear you.
I'm sorry.
It's okay. So these cold sore viruses are herpes viruses. Now you have 9 different herpes viruses that infect people, and 7 of them are naturally you're born with. You get from your parents, right? And they're the ones that kind of cause cold sores when you get sick, right? And 2 of them you are the ones you acquire. One is sort of you can acquire from like kissing someone with herpes. The other one is a sexually transmitted version. So these are viruses though, and they cause these cold sores as a result of their infecting your cells. And then you've got these respiratory viruses, like respiratory syncytial virus, which is really bad for kids. It's actually a big problem in many countries with kids that have respiratory challenges, respiratory syncytial virus, and we don't really have any cures for it. And then you've got viruses that infect the immune system, like HIV, human immunodeficiency viruses. So these are some of the common virus infections that we're battling today. And one thing I wanted to really point out to you guys, and this is great stuff for you guys to look up. Viruses occur in families. There's a whole kind of, taxonomy to identifying viruses. There's actually an organization that's dedicated to identifying and classifying viruses, just like we do with animals and other species of plants around the world. We can classify viruses. And viruses come in 3 different kinds. They're DNA viruses, and these are some examples of DNA viruses like pox virus, herpes virus, adenovirus, and some of the hepatinoviruses are DNA viruses. And their genomes are double-stranded DNA, much like our chromosomes are double-stranded DNA, right? They have different kinds of capsid structures, which make them look different. But so this is like a picture of an adenovirus. It looks like it's a very kind of geometrical shape, and it's got these little spikes sticking off the end of it that it uses to get inside a cell. So you can see these different viruses in solution. Vaccinia virus is big. It looks like a big round oval, and it's kind of like it's got a bunch of proteins on its cells, so that makes it look kind of wrinkly there. And then herpes simplex virus, again, looks sort of like adenovirus. It has more of a structure, a geometrical structure, because these are the proteins of the viruses that aggregate according to these geometrical structures many times, okay? So you've got these DNA viruses that can cause different kinds of diseases, mostly cold sore type of diseases or even the common cold, right? Then you have some nastier viruses. So most of the nasty viruses, like the viruses that really cause problems in us, are what's called RNA viruses. So these viruses have skipped the DNA phase, right? So if I go back to the DNA viruses, these DNA viruses encode the nucleic acid to make themselves over and over again. And that DNA gets converted to RNA, which gets converted to the protein. So they use the host machinery that we use every single day when our cells are replicated to take DNA to RNA and protein, right? These other really bad viruses that really can kill us and replicate really fast, right? They just skip that DNA step altogether. They use RNA. And there are 2 types of RNA viruses. One is called a negative sense RNA virus, and the other one is called a positive sense RNA virus. So we're going to talk about both of them. I'm going to start with positive sense, right? So positive sense means that you go from 5' to 3'. Remember that from our PCR studies? 5' to 3', the sense strand, right? It literally means they make an RNA strand that encodes all of the proteins for the virus. So in the case of coronavirus, its genome is about 30,000 letters, A, U, G's, and C's, right? Because it's RNA. So A, U, G's, and C's that are 30,000 letters that encode about 10 of these genes, and that make all of the genes required to make the virus particle. And then all it does is it injects itself into your cells and replicates like crazy until it kills your cells. And in this case, it kills your lung cells, the coronavirus. Other viruses like rubella, hepatitis C, same kind of thing. Positive sense RNA virus. That means they function very much like the RNA that's in your cells, and they can hijack your machinery to make more of themselves. Okay. The negative sense RNA viruses are RNA, but just in the opposite direction. So they start off 3' to 5', then they have an enzyme, right, called an RNA-dependent RNA polymerase that makes them go from 5' to 3'. So their genomes are in the reverse orientation, and then they have to, once they get into the cells, they make the positive orientation and to make more of themselves. And these viruses can replicate really fast, like Ebola and influenza. These are some of the fastest replicating viruses in cells, right? So they can replicate within an hour. They can get inside a cell and completely wreck it. So they can do it millions at a time. So measles, rabies, Ebola, influenza, some of these other viruses like Lassa and hantavirus that cause deadly diseases are all these negative sense RNA viruses. Okay?
Just out of curiosity, what does a Hanta virus do?
Hantavirus is a virus that can cause encephalitis in people and kill you. And it's spread from different kinds of mice families in their droppings. And it's a really nasty virus. These other kinds of viruses called Nipah valley fevers, a lot of these kinds of viruses are found in Africa and other countries because they have natural animal hosts that these viruses grow in animals, right? But they don't kill those animals. Those animals have actually, negotiated a good relationship with the virus so they don't get killed. But when they get into humans, because we haven't seen them before or haven't seen them much, they kill us pretty bad.
I have another question.
Mm-hmm.
How does a virus mutate?
So that's a great question. So when a virus replicates, and before I go into COVID, let me try to address that. I wish I had a better diagram for it. But remember how our cells make their DNA, replicate DNA using the DNA polymerases, right? Mm-hmm. And then you can make RNA using RNA polymerases. These viruses have proteins called polymerases, right? That's what they do. They actually make more RNA. So they use, once they get into your cell, and you've got all of the ingredients to make RNA in the cell because you need it for your own cells. That's why viruses, they just come in and hijack all of the cell's machinery to make more of themselves, right? So they use these enzymes, actually proteins, that the viruses encode called polymerases. And then their polymerases are not really good at fixing errors. Like when our cells replicate, we have our DNA polymerases have like a whiteout, or they can fix the error. So if an error is made, let's say an A becomes a G, many times the DNA polymerase can sense that, our DNA polymerase can sense that and go back and fix it. Sometimes it doesn't, and you get a mutation that can lead to a disease. But in the viruses, they don't have the correcting mechanism. They have no whiteout. So they just make an error and just keep the error. And then what happens is it winds up mutating the virus. And then what that does for the virus is it allows it to explore all the different sequences it can make that can make it best suited to replicate in the cell. And it also is a defense for them because just as fast as the virus is changing how it looks, our immune system is changing what antibodies we throw at it, right? And so it's a really complex race between the immune system and viruses. And, sometimes we win, sometimes they win. Does that make sense?
Mm-hmm.
Okay. So all right, so wrap up, and I wanted to talk. Let's get to fast forward to the current last 3 months. Coronavirus, COVID-19 outbreak. Notice it's called coronavirus 2, SARS-CoV-2. And it's a new type of coronavirus that was detected in China maybe about 4 or 5 months ago. Actually, in October was when they first found this in bats.
I have a quick question, Dr. Cunningham.
Yep.
And were they eating the bats? Oh.
Yeah, it's unclear whether they were eating the bats or if they were just somehow in contact with these bats, like bat droppings or other things. And then somebody got this bat virus in them, and then it mutated to the point where it
Dr. Cunningham, I think you covered your microphone.
Yeah, you covered your mic with like your hand.
Oh, I'm sorry. Can you guys hear me now? Can you hear me?
Yeah, we're good.
Yep. So let me say that again. So it came from bats, right? And it may have actually gone into another species called a pangolin, It's like a armadillo-like animal over in China. And then it got into humans somehow. They don't really know how that first transmission event occurred, but somehow these folks either had contact with these bats in these exotic food markets, or just someone had contact with the bats just, in nature, like, had come into it and come in contact with them and then acquired the virus. And then when they got to Wuhan, China, they began to spread it to people in Wuhan, because then it became, you had patient 0, which was the very first person that could spread that bat virus. And then it became a human virus once it got spread from that first person to the next person. Okay. And this human version of that bat virus is now what's spreading and killing everybody now. Okay. And so far, as of yesterday, we have 627,000 people that have been infected, and 28,000 people have died from this disease. And almost every country on the face of the earth, 192 out of 195 countries, have reported at least one case of this virus there, right? And here's the symptoms that it causes, right? Most people are actually going to be okay. The actual death rate for that virus you can calculate. What can someone divide for me? What's 28,714 by 627,327? What's that number? I'll let you guys tell me.
I got 3%.
3%. So that's your mortality rate. That's your death rate for this virus. So a global death rate of about 3%. Now, if you compare that to influenza, the global death rate is about 0.1%. Okay. So this virus is 30 times more deadly than influenza. But if you compare it to Ebola, the death rate is 30%. So it's 10 times less deadly than Ebola, but it spreads about 1,000 times faster because Ebola is not airborne. Ebola only spreads from person to person touching, right? So that's why the outbreaks are easier to contain.
So when they first tried to vaccinate people for the virus, do you think they tried the vaccination that they used for the SARS virus, since they're both similar?
Well, that's just it. They did, but they're different enough where it doesn't seem like the SARS virus CoV-1, the first version of SARS virus that happened in early 2000s, they outside of it, these red proteins out here. You see this picture of the virus? Can you guys see this?
Mm-hmm.
I'm sorry. Oops, I don't know what happened. Oops, let me go back. You see this picture of the virus?
Another one.
These little spikes, right? These are the things that these are the envelope proteins called the spike glycoprotein that your antibodies recognize. Now, the ones for COVID-19, the SARS-2, look different from SARS-1. So the vaccines for SARS-1 won't work against SARS-2. Okay? So they're trying to make a new vaccine, and they're really rushing to make a new vaccine by making something literally that looks like coronavirus but can't be infectable. Okay? And these are, so what happens is this virus gets into your lung cells, and just like the smallpox virus caused those plaques and kills those cells, it's causing plaques in your lungs, and it destroys the tissue in the lungs. And that's why in 3% of people that have a really bad infection of this, that it kills them. And most of the people that it's being fatal in are people over the age of 65. Now, why do you think that is?
They have weaker immune systems.
That's right. So they have less of a chance of their immune system creating the antibodies that would stop the virus infection and prevent it from destroying the lungs.
So those who survive the virus come out with permanent lung damage?
Some of them. Some people seem to have lost lung function. Sometimes, 20%, 30% lung function. But some other people can be completely asymptomatic. Now, what do you think makes for these differences in how people are responding to the virus?
How healthy they are.
And what else?
Their genes.
Their genes. That's right. Their personal genetics. And so we're doing actually an intellectual study right now looking at people's genetics in terms of the protein that the virus uses to get in. There's a protein that the virus uses to attach to the cells. It's called ACE2, ACE2, okay? And it's angiotensin converting enzyme. It's an enzyme that's actually on the surface of cells. And the SARS spike protein, this red thing right here, binds to ACE2. And when it binds to ACE2, it allows another small protein, this little orangey protein on there called the E protein, to bind to another protein on the cell surface called TMPRSS2. And then TMPRSS2 is what's called a protease. It actually cuts it, it cleaves it, and allows the virus to get into the cells. So it's a very complicated way that it gets in. And certain people have healthy immune systems. They have healthy immune systems, can make antibodies that can block all that and stop it from happening. And they can shut down the infection. And depending on what the genetics of your ACE2 are, you might have some natural immunity to it. So I think some percentage of people might be naturally immune to it. So it's going to be very interesting for scientists to track, look at that kind of information, and see, which people are most susceptible.
Dr. Cunningham, what I don't understand is that some people have reported that they don't have like a sense of taste or smell once they're treated with the coronavirus. Why is that?
That's a good question. I don't think we really know that yet. My hypothesis would be that some of the cells that are responsible for transmitting the signals for taste and smell are getting disrupted by the virus. So the sensations of taste and smell are due to neurons that transmit a signal from your tongue, right? The cells in your tongue, and then from your nose, your nasal. You have nasal neurons that extend into your cavity. So those to your nasal cavity. So those neurons may be being affected or infected by the virus and getting killed, and that's probably why you lose your taste and smell. You lose your sense of taste and smell. Will
they eventually come back, or is that?
They will, and I think when the virus infection passes, then you probably regain those sensations. Sorry if I covered the microphone.
Can you get the virus twice?
It doesn't appear so, because just like when you recover from a cold, you're not able to be infected with that strain of cold virus again. It doesn't seem like people that have recovered from COVID-19 are then being reinfected by it. But honestly, it's so new, we haven't studied that yet. I think that's an active area where researchers are going to be looking for. Because if it turns out that you can get reinfected, then that tells us that our immune response is not really that great. So we're going to have to find things like drugs that can block the virus at multiple steps, just like we've done for HIV.
Okay, so doctors and everybody else are completely new to this virus. So it's going to take about a couple of years to come up with a vaccine. Is that right?
Maybe 18 months. I think we're going to see literally how fast can we make a vaccine as a society. They're pushing it as fast as they can. And I was on a phone call with scientists from around the world when this outbreak first occurred, that my PhD advisor, he was one of the conveners of it. And I was just listening in. They're trying to do everything possible, because I just wanted to understand what was the current status in China. And they are trying to make a vaccine as fast as is humanly possible. But there's some concerns. There's not, with any new vaccine, two things. One, you don't never know how well it's going to work, right? Until you get it out there. And then two, in some types of respiratory diseases, let's say like for respiratory syncytial virus, which is another respiratory disease, sometimes the vaccines actually make the infections worse. So one of my concerns is that some people, it might make the infection worse because you can make these antibodies that help the virus get in. There are viruses that can block the virus getting in, and then there are also some types of antibodies you can make that can help the virus get in. And the people that are also dying faster from this could be making those kind of antibodies too that could worsen their disease. There's still a lot we don't know. And so I think there's going to be a lot of scientific research on this. And our goal is to really fund a lot more research. I hope our government funds a lot more research on this so that we're prepared for the next outbreaks.
Can't you develop pneumonia from this?
You can, because it destroys your lung tissue. Mm-hmm. Anything that destroys your lung tissue can cause you to get pneumonia, which is essentially fluid being released into your lungs till you drown in your own lungs. So let me stop here. So I'm going to show you guys some stuff. And actually, when you come to the lab, I talk about HIV a lot because HIV is one of the viruses I studied. And for my PhD, I was actually studying how it moves around inside of cells. And when you ask me how we see viruses, using molecular biology, I was able to to put in a green fluorescent protein into HIV and then put it on cells in a microscope so I could see it. And I'm going to see if this video will play. Okay, it's not going to play because the media is not there. But this is on YouTube. I'm going to show you guys this video. So if you Googled my name and HIV, this will come up, this YouTube video. And it's showing a green fluorescent protein labeled HIV on live cells. And I used this because I was having to figure out how HIV moved around inside of a cell. So I had to actually look at it. And because you can't see viruses with a naked eye, I had to make a way to see it by putting like a colored tag on it. So I made it glow in the dark, literally, so I could see it. So it's pretty cool. And so, yeah, I mean, this virus, this is what it looks like. So coronavirus is not much different. It's around the same size, but it has a longer genome. HIV has about 10,000 base pairs. Coronavirus has 30,000. And these are both RNA viruses, except for HIV has 2 copies of RNA. The coronavirus RNA is just one long circle that stretches in among the membrane, okay? But it makes these particles that then attack your cells. And its goal is to just make more of these particles, keep attacking your cells until they burst. And that's what happens. Okay, and so I wanted to show you guys what we do in the lab is using PCR, we can get the sequence of the virus. These are the genes. These are the genes of the virus this is the genes of the HIV virus that my other PhD thesis advisor, when he was maybe 25, he sequenced this virus for the first time. And back then, it took him an entire year to sequence the virus, working nonstop and only sleeping 3 hours a day, right? And then once he did that, now we had the sequence of the virus proteins, so we can start to design drugs for the virus proteins, like the reverse transcriptase that's in the polymerase gene, like the protease that is a protease inhibitor. And then you've got these 2 kinds of drugs that attack the reverse transcriptase and the protease are part of the cocktail. And so using that sequence information, researchers were able to design drugs to these proteins that made the virus shut down. And so that's exactly what these same researchers are going to try to do for coronavirus. Back then, I told you, it took them about a year working nonstop to get the sequence of this virus. Today, we were able to get the sequence of this coronavirus within 2 weeks of being discovered, okay? So I have the sequence of coronavirus on my computer now, and I'm looking at it and looking for weak spots on it, and I'm contributing my thoughts to scientists all over the world to try to find some drugs for it. And that's what we want to do. But this is a diagram of what actually happens when viruses infect a cell. This is what HIV does. The virus attaches to the cell. That's the first point where you could block it. Then it gets into the cell. Then once it gets in, it has to make more of its genetic material. Now, HIV does it a different way. It actually takes RNA, makes it into DNA, integrates into your DNA, and then makes it RNA again. That's why it's called a retrovirus, because most viruses just go straight to the RNA and make protein, okay? That's what coronavirus does. But all of these steps are places where you can design drugs to block the virus. You can block the virus coming in, you can block the virus after it gets in, you can block the virus as it's trying to get out, and you can block the virus even after it's gotten out to keep it from making a mature particle that can infect. And so we're gonna try to hit coronavirus at all these stages, just like we did for HIV, and make a cocktail of drugs that can block it. And these are all the different drugs that were made for HIV. And this is how we develop drugs, right? I'm just showing you, it took, let's say, 10 years to go from the first drugs to the drugs that became the cocktail. And I don't think it's going to take us that long with coronavirus because we have literally everybody around the world working on this. So I'm hoping that we can really move fast and get some new things out there. All right? So I'm going to stop the lecture there. And then I'm going to have some questions from you guys. What questions do you guys have? Everybody can unmute.
So I saw an article title. I didn't read the article. I just saw it before joining this. And they found that coronavirus patients in China who tested positive and recovered are testing positive again. And I just wanted to see
That's scary. Then that's something that we're going to have to say, because then what that says is that when they recovered, they are now being infected with a new version of that coronavirus that they cannot protect themselves from with the existing immunity. So that's a scary thing. So that says to me that the vaccine might not be as effective as we need it to be.
So like, would that make the vaccine like harder to make or like longer to make?
It can make a vaccine harder to make and harder to have what's called like universal protection, right? From a vaccine, you really want to have universal protection. You want to have the strain that you make of the vaccine be able to protect millions of people. Now, if people who have coalesced, that means people who have actually recovered from the virus, are then being reinfected with new strains of the virus. Very scary. So we're going to have to find drugs. Other questions? Let's hope that's only isolated to a few number of people. And we'll have to see, man. I mean, I think we're still early in the outbreak. But one of the questions I have for you guys is, knowing what you know, how would you go about trying to find drugs for this virus? What would you do?
Well, so we have a similar part, COVID, SARS-1. So we would probably go there for like a basis and like how we find future drugs, right?
Yeah, I think that's a good place to start, is look at drugs that had been found to at least be somewhat effective against SARS-1, and then start there, and then screen a bunch of drugs against SARS-2. You know what it means to screen a drug? Test it. That's right. So people like us, molecular biologists, we make up the test in a test that people use, that we can now use to screen a bunch of chemical compounds that are drugs and see which ones can stop the virus. So remember that picture I showed you of that HIV virus that was green? I could use that as a screen now, because I can see all of the virus infected cells would be green. If I had a drug that blocked them, there would be no green. So I have a plus and a minus signal.
Also, I remember my teacher mentioning, is it true that we have like advanced technology enough that you just put in a genome sequence and it like spits out recommendations for vaccines?
Yes, you do. You have computational biology now that you can predict what parts of a protein from a virus the immune response will likely respond to. And then you can have researchers make what's called peptides corresponding to the amino acids for those parts of the protein, the viral proteins, and then see if those work.
That's really cool. You can do
it's super cool. Now. It's in silico biology. I think it's the coolest thing. And you can use things like artificial intelligence and machine learning to really refine those methods. So it's a really interesting mix of computational techniques and biology.
I have a question. So when it first came to America, I remember there were people on the cruise ship who were tested positive for coronavirus, but there weren't others that were tested. Do you believe that we accidentally infected other people who weren't tested positive? And do you think we made that mistake?
Probably. I think a lot of mistakes were made early on. I think our government made a lot of mistakes early on. They didn't take the outbreak seriously enough, and they didn't do enough testing early enough.
So then when people were quarantined and who didn't have it, but the ones who did have it, they kind of mixed them up so that everyone else had it. Is that right?
Yeah, and that's a big mistake. With this virus, because it spreads so fast from airborne or also on surfaces, because it can live for 3 days on a surface, right? At least 3 days, right? In some cases, on some of those cruise ships, they were screening areas for like 2 weeks and still finding coronavirus 2 weeks later. So it can stick around for a while. That's why everyone's saying wash your hands, wash your hands, wash your hands, and try not to touch your face, because that's how viruses get in. When you touch your face and your nose and your eyes and stuff, that's how viruses get in. They're so small you can't see them. That's why you got to constantly try to clean your hands.
I love washing my hands.
That's good, Ahmad. Good practice. And now that's why we're doing these social distancing protocols. Our only hope to stop the spread of the virus in this country right now, because we don't have a vaccine and we don't have any medicine for it, is to stay away from each other. And it's probably going to happen for a few months, because you have to have the people who are carriers. Because one of the other concerning things about this virus is that there are people who can be carriers that don't have any symptoms. Mm-hmm. So you literally don't know who's infected. And if you are what's called an asymptomatic carrier, that is, you have the virus, but you're not getting sick from it, you could pass that on to somebody who's at risk, like an older person, and it could kill them. So that's why it's really, really dangerous right now for us to have contact. That's why we have to take these really drastic measures. Now, we're not even being as drastic as China was. China, my friend is from Wuhan, China, who has family there. And he was telling me that his family had basically been locked in their homes pretty much for 2 months. The government was bringing them groceries. They weren't allowed to leave their buildings. They weren't allowed to drive. The streets were closed. It was complete lockdown.
Isn't Italy doing the same thing?
Yeah.
Italy's trying to do the same thing, but it's harder in our societies because our governments are different. You know, China, they have more of the military can just impose more of their will on the people. Like, the people kind of have slightly less rights in that regard. Here, we are used to our freedoms. We're used to being able to move around, and that is going to make it very hard here. Plus, you know, I think the leadership that you've seen out of the or lack of leadership out of the executive branch has been challenging, and so that's why states are having to step up and governors are having to step up and really do the right thing.
Is the virus heat resistant?
No, you can kill it with heat. You can kill it with alcohol. If you heat it up over 100 degrees, it will bust apart just like anything that has DNA or RNA.
Clorox wipes.
Do you think it will slow down in the summer?
Clorox wipes and 70% alcohol. That remains to be seen whether it'll be slowed down by heat, because what it is that we don't know that necessarily that the temperature change will slow it down. It may kill it on surfaces more in the hot weather, but the person-to-person spread may still happen because it is respiratory viruses. They tend not to spread that well in summer months because people aren't inside and around each other as much. When you think about it, winter months, everybody congregates indoors. We stay in close quarters. You know, we're breathing the same recycled air. But in summer months, everybody spreads out. We get out more. We spread space out more. So I think I'm hoping that by like June, July, you're going to start to see it go down a bit.
Do they advise like washing your hands instead of hand sanitizer, because like hand sanitizer doesn't have enough alcohol in it?
Yes, you need to use a hand sanitizer that has at least 70% alcohol. Okay, 70% alcohol will kill most things. That's what we use in the lab to sterilize stuff. Okay, so when I teach you guys cell culture, you're going to see like I spray everything down. I have a spray bottle of 70% alcohol. That's literally 7 parts alcohol, ethanol, and 3 parts water. And then I spray everything down with that to sterilize it. That kills all bacteria, viruses, all kinds of stuff.
Yeah, because
Clorox wipes will do the same thing. So for our next sessions, I think what I'd like to do, actually what I want you guys to do before our next session, each one of you, is to come up with your own idea of how you would try to find a drug for coronavirus.
Gotcha. Let me write it down before I forget it right now, because I already have an idea.
Yeah, me too.
And then we're going to share our ideas, and then we're going to pick one and see if we can't do the intellectual exercise of designing the experiments for it. Sound good?
Mm-hmm.
Yeah.
Yeah.
What's up, George?
All right. So I'll share these slides with you guys. These are very kind of complicated slides. You normally wouldn't get this kind of lecture until college. So you guys have just now had a college lecture basically, but it's good background reading for you as you're reading up stuff. Now, if you want to learn more about viruses, there's one book I recommend called Fields Virology.
Fields Virology.
F-I-E-L-D-S. Fields Virology. It's literally the encyclopedia of all viruses. I had to basically memorize that book when I was in graduate school getting my PhD.
What was that other book you told?
The Molecular Biology of the Cell.
Okay, yeah. There's like different versions. There was like a do you want us to get the latest version or the?
Yeah, get the latest version.
Okay.
I'll tell you what's the latest edition. I'm looking it up right now.
You said Fields virology.
Yeah, Fields Virology. Let me see. I'm just trying to think what the last edition is of it. They make a new one every year, basically. It's gonna tell me in a second. Let's see. I think it's up to the 6th edition.
Okay.
Yeah.
For the Fields virology, who's it written by? Because there's like different versions of it.
It's written by Fields. Let me get mine. Hold up. Where's my virology book? Oh, it's upstairs because I was teaching my kids it. Hang on. I'm gonna travel around my house a little bit so I can go find my virology book.
Virtual tour.
Written by Fields. The guy's name is last name is Fields. That's what they call it, Fields Virology.
Mm-hmm. Oh wait, Nip is it?
I'll look at his first name. Hold on, I'm gonna look on my copy. It is by Bernard N. Fields and David Knipe.
Okay, yep.
Yep. So it's also known as Fundamental Virology. I mean, it's the title of the book, but a bunch of other professors contributed to it. And I think it's up on the 6th edition.
That's like $500.
Yeah, and you can see it online. Don't pay for it, but you can see some of it online, I think. But that's only if you want to learn more about it. I have some other books and stuff. And then you can also Google these things. So some of the slides that I have, if you want to learn about more viruses, I just want you guys to know that they come in these different families, right? So like, actually I was going to show you guys something because this was a paper. So this is what I do in my spare time. Can you guys see my screen still? Hang on. Am I sharing my screen still or not? Let me see if I can reshare my screen. I'm going to share my screen again, okay? I'm going to show you guys something, okay? I'll show you guys something. Okay. Can you see this?
Mm-hmm.
This is actually the paper, the first paper where they published the sequence of the new coronavirus. Okay? It's a group of Chinese researchers that they identified it on January 5th. So within 15 days, right, they had the sequence and were able to publish it. And then they could tell what family of virus it's related to. This is called a phylogenetic tree right here that shows how closely related this coronavirus is to the bat coronavirus. That's how they knew it came from bats, because they're comparing the sequences of these different viruses. And these bars, if they're close together, it means the sequences are very close together. You guys hear me? Yeah. And you see, this is the genome of the coronavirus. It's 30,000 nucleotides. And these are the proteins that it makes. So each of these proteins, like this S protein, was that red spike protein on the outside of the virus. So this would be a good target for drugs. And you can, that's another open reading frame that are these things called ORF. It's called an open reading frame. These are just literally long polypeptides that the virus protease cleaves up to make the smaller proteins of the virus, to make the whole thing. But this is a good paper. I'm gonna send you guys this. It's complicated, but if you can understand it, I'd be very impressed. This was when they were doing experiments to see which kinds of cells could be infected with the virus. And so this receptor on the cells is called ACE2. It's a protein on the surface of cells. And they looked at proteins, different kinds of ACE2s from humans, from bats, from different things to see if they could be infected. It's a very good paper that they turned around in 2 weeks. So I'm going to send you guys this just for fun. And you can tell me if you understand it. We can discuss it the next time. Okay?
Mm-hmm.
All right. Okay, that's all I had today. Did you guys have any other questions and stuff?
Nope.
No.
I'm gonna keep going with your other project too. So the next phase with that was to actually make the virus, make the vector virus in cells, and so I'm gonna try to do that in the lab as soon as our labs open back up. Okay.
Okay.
Hopefully before the end of the year. You can do that. Say hi to my guys. They missed you guys.
Hey guys.
Boys.
Hey.
Okay. Well, if I don't have any other questions, you guys did great. So I will I'll talk to you guys, I guess, in another couple of weeks. And so, you know, your assignment is to come up with your idea, and then we'll discuss it during our next session.
All right.
All right. All right. You guys stay safe. Keep your parents safe. Keep your distance from everybody, and keep reading. All right.
To help us with our biomedical sciences program, we ask that you go to future-kings.org and make a donation.